8.5.3 Endomembrane system. While each of the membranous organelles is a distinct in a terms of its structure and function, many of these are considered together as an endomembrane system because of their functions are coordinated. The endomembrane system include the endoplasmic reticulum, that is ER, Golgi complex, lysosomes and vacuoles. Since the functions of the mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered as a part of endomembrane system. 8.5.3.1 The endoplasmic reticulum, that is ER. Electron microscopic studies of the eukaryotic cells reveal the presence of the network of a reticulum of the tiny tubular structures scattered in a cytoplasm, that is called the endoplasmic reticulum, that is ER, figure 8.5. Hence, ER divides the intracellular space into the two distinct compartments, that is luminal that is inside ER and the extraluminal, that is cytoplasm compartments. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called the rough endoplasmic reticulum, RER. In the absence of ribosomes, they appear smooth and are called the smooth endoplasmic reticulum, SER. RER is frequently observed in the cells actively involved in a protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site of synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER. 8.5.3.2 Golgi apparatus. Camillo Golgi 1898 first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1.1 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with a distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus are principally performs the functions of packaging materials to be delivered either to the intracellular targets or secreted outside the cells. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in a close association with the endoplasmic reticulum. The number of proteins synthesized by the ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus. Before they are released from its trans phase, the Golgi apparatus is important site of the formation of glycoproteins and glycolipids. 8.5.3.3 Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in the almost all types of hydrolytic enzymes, that is hydrolases, lipases, proteases, carbohydrases, etc. optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and the nucleic acids. 8.5.3.4 Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product and the other materials not useful for the cell. The vacuole is bound by the single membrane called the tonoplast. In the plant cell, the vacuoles can occupy up to the 90% of the volume of the cells. In plants, the tonoplast facilitate the transport of the number of ions and other materials against the concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for the osmoregulation and excretion. In many cells, as a protist, food vacuoles are formed by the engulfing of the food particles. 8.5.4 Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is a variable depending upon the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is a sausage-shaped or a cylindrical having a diameter 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into the two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix, and the outer compartment forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called cristae, singular crista, towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial functions. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouses of the cell. The matrix also possesses the single circular DNA molecule, a few RNA molecule, ribosomes, 70S, and the components required for the synthesis of proteins. The mitochondria divide by the fission. 8.5.5 Plastids. Plastids are found in all plant cells and in the euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into the chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for trapping the light energy essential for the photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophylls, and the other are present. These give plant a part of the plant to a yellow, orange, and a red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. Amyloplasts store the carbohydrates, means starch. Example, potato. Elaioplast store oils and fats, whereas the aleuroplast store the proteins. Majority of the chloroplast of the green plants are found in the mesophylls of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having a variable length, means 5 to 10 micrometer, and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limiting by the inner membrane of the chloroplast is called the stroma. The number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like piles of coins, called grana or intergranal thylakoids. In addition, there are the flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclosed a space called lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains the small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, that is 70S, than the cytoplasmic ribosome, that is 80S. 8.5.6 Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as a dense particle by the George Palade. They are composed of a ribonucleic acid, means RNA, and proteins, and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80S ribosomes are 60S and 40S, while that of the 70S ribosomes are 50S and 30S. The S, means Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of the density and size. Both 70S and 80S ribosomes are composed of two subunits. 8.5.7 Cytoskeleton, an elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilaments, and the intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shell, maintenance of the shape of the cell. 8.5.8 Cilia and the flagella. Cilia and flagella are hair like outgrowths of the cell membrane. Cilia are a small structure which work like oars, causing a movement of either the cell or a surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possesses a flagella, but these are structurally different from the eukaryotic flagella. The electron microscopic study of the cilium and the flagellum show that they are covered with the plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has a 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of the axonemal microtubules is referred to as the 9 plus 2 array. The central tubules are connected by a bridges and is also enclosed by the central sheath, which is connected to one of the tubules of the each peripheral doublets by the radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and the flagellum emerge from the centriole-like structure called the basal bodies. 8.5.9 Centrosome and the centrioles. Centrosome is an organelle usually containing the two cylindrical structures called centrioles. They are surrounded by the amorphous pericentriolar materials. Both the centrioles in an centrosome lie perpendicular to each other, in which each has the organization like cartwheel. They are made up of the 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplet are also linked. The central part of the proximal region of the centriole is also proteinaceous and are called the hub, which is connected with the tubules of the peripheral triplets by the radial spokes made up of protein. The centrioles from the basal body of cilia or flagella and the spindle fibers that give rise to the spindle apparatus during the cell division in animal cell. 8.5.10 Nucleus. Nucleus as a cell organelle was first described by the Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given name the chromatin by Fleming. The interphase nucleus of the cell, when it is not dividing, has a highly extended and elaborate nucleoprotein fibers and chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of a two parallel membrane with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears the ribosomes on it. At the number of places, the nuclear envelope is interrupted by a minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which the movement of the RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei also frequently observed. Can you recollect the names of organisms that have more than one nucleus per cell? Some mature cells even lack the nucleus. Example, erythrocytes of the many mammals and sieve tube cells, vascular plants. Would you consider these cells as a living nucleus? Example, erythrocytes of many mammals and the sieve tube cells, vascular plants. Would you consider these cells as a living? The nuclear matrix or the nucleoplasm contains the nucleolus and the chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in the cells are actively carrying out the protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during the different stages of cell division, cells show the structured chromosomes in a place of nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of a DNA distributed among the 46 means 23 pairs of chromosomes. You will study the details of DNA packaging in the form of chromosome in the class 12. Every chromosome that is visible only in the dividing cells essentially has a primary constriction or a centromere on the besides which disc-shaped structures called kinetochores are present. Centromere holds the two chromatids of the centrosome. Based on the position of the centromere, the centrosomes can be classified into the metacentric chromosome has a middle centromere forming the two equal arms of the centrosome. The submetacentric chromosome has the centromere slightly away from the middle of the chromosome, resulting into the shorter arm and one longer arm. In case of the acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one extremely long arm, whereas telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have a non-staining secondary constrictions at the constant location. This gives appearance of a small fragment called the satellite. 8.5.11 microbodies. Many membrane-bound minute vesicles called the microbodies that contain the various enzymes are present in both plant and animal cell. Summary. All organisms are made up of cells or aggregated of cells. Cells vary in their shape, size and activities functions based on the presence or absence of the membrane-bound nucleus and the other organelles, cells, and hence organisms can be named as a eukaryotic or a prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus and cytoplasm. Plant cells have cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitate the transport of the several molecules. The endomembrane system includes Golgi complex, lysosome and vacuoles. All the cell organelles performs a different but specific function. Centrosome and a centriole form the basal body of the cilia and flagella that facilitate the locomotion. In animal cell, centrioles also forms the spindle apparatus during the cell division. Nucleus contains nucleoli and chromatin network. It is not only controls the activities of the cell organelles but also plays major role in the heredity. Endoplasmic reticulum contains the tubules or cisternae. They are two types, rough and smooth. Helps in the transport of substances, synthesis of proteins, lipoproteins and glycogen. The Golgi body is membranous organelles composed of a flattened sacs. The secretions of the cells are packed in them and transported from the cell. Lysosomes are single membrane structures are involved in the protein synthesis. These occur freely in the cytoplasm or are associated with the Mitochondria helps to oxidative phosphorylation and generation of the adenosine triphosphate. They are bound by the double membrane. The outer membrane is smooth and inner membrane one folds into the several cristae. Plastids are pigment-containing organelles found in the plant cells only. In the plant cells, the chloroplasts are responsible for the trapping light energy essential for the photosynthesis. The grana in the plastid, it is the site of a light reactions and the stroma of a dark reactions. The green-colored plastids and the chloroplast which contain chlorophyll, whereas the other colored plastids are chloroplast which may contain pigments like carotene, xanthophyll. The nucleus is enclosed by the nuclear envelope, a double membrane structure with the nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, the cell is the structural and functional unit of life. Thank you.